Good afternoon, everyone. May I have your attention? It takes usually a few seconds. Our speaker today is Maureen Lynch, who is currently a postdoctoral fellow in biomedical engineering at Cornell. She received her Bachelor of Science degree from Clemson University, as well as an MS and a PhD degree from Cornell in mechanical engineering in 2008 and 2010, respectively. Maureen is also a faculty candidate. Please welcome our speaker.
Okay, thank you all for coming to hear me talk about my research. Can you all hear me okay in the back? Okay, fantastic. Yeah, I guess I can't really see if that was a thumbs up or a thumbs down, so if you can't hear me, shout. Again, thank you for having me here to talk about my research. It's been a really great visit. I'm pretty excited about Dartmouth, so hopefully you guys enjoy my talk. What I'm going to talk to you about today is going to focus on my research from my postdoc, and I'll touch on a little bit of my work from my PhD. So today I'm going to talk to you about how mechanical loading decreases osteolysis and tumor formation via bone remodeling changes. This is the overview of today's talk. I'll give you the clinical motivation for this problem, the experimental approaches that I use, and then I'll finish up with some conclusions and future directions. So the clinical problem that my work is trying to address is breast cancer bone metastasis. The skeleton is the most common site for breast cancer metastasis, and it's also true from several other cancers like prostate cancer. 3 in 4 patients with advanced breast cancer will develop incurable bone metastasis. And this metastasis is osteolytic, which means it's bone degrading. So if you'll draw your attention right here to the CT scan, sorry you guys, you can't, hopefully, let me use the mouse. If you draw your attention right here, you can see that there's a very large lesion. And you can appreciate right away that the tumor occupying the space is large, that the hole that it leaves behind in the skeleton causes a lot of fragility, which means risk for fracture. And then, of course, there's pain. So overall, the prognosis for patients is very poor. So again, this is the clinical problem that we're trying to address, that I'm trying to address with my research. Before, to touch on how this cancer affects the normal bone environment, first let me tell you what normal bone remodeling is. This is important because cancer cells take advantage of normal processes that are going on in our body, and this is true for the skeleton. Cancer cells take over the bone remodeling process. So bone remodeling consists of primarily two types of cells, osteoblasts. The mnemonic device for you to use is BLAST, which is B for build. So these cells build bones. And the other cell type is the osteoclast, which tears down bones. Let's give this a go. Here we go. Osteoclasts, osteoblasts. So first, progenitor osteoblasts are around in your skeleton all the time. So if there's a need for some remodeling to happen, these progenitors will recruit pre-osteoclasts from circulation and contribute to their differentiation and ultimately their resorption of a little packet of bone. And after that, the osteoblasts, B for build, osteoblasts will come into the same place, though it's not here in my cartoon, it happens in the same place. These cells will lay down new bone. So normally, when in a healthy individual, the relative balance of these two cells and their activities are balanced. And that means that our bone mass stays the same. We don't lose mass. And this is the way in which we keep a healthy skeleton. If you have some sort of a little fracture, remodeling will take care of that. If you have a need for calcium, your remodeling will take care of that. And lots of things can shift the balance between these two cells, like some of the things I just mentioned, such as a need for calcium. But also the two things that I'm interested in, mechanical forces will change the balance, and then breast cancer that has metastasized to the skeleton will change the balance. So to drill in a little bit deeper to what's happening once these cells get to the skeleton, let me tell you about the two types of bone that people usually use to discriminate their work. So when I tell you skeleton, when I tell you bone, you are probably thinking about this very dense cortical bone, which makes up the shaft portion of our long bones. But there's another kind called cancellous bone, and that's the porous bone that's found at the end of your long bones, and it's found in your spine. We care about this kind of bone because this is the place where if anything is shifting the balance between those two cell types, if anything is shifting the bone remodeling balance, this type of bone is disproportionately affected. This is because bone remodeling is a surface-mediated process. You can see right away this is high surface area to volume ratio. So that means if there's a shift towards bone loss, this is the site where bones are going to fracture. And then it's also the site where breast cancer cells colonize when they've arrived in the skeleton. So it's this uniting of these two biological features in the same, I guess I'll call it anatomical place in the skeleton. So once the tumor cells get there, what happens? Well, as I said, ultimately they cause bone degradation. So the cancer cells come in and they basically get these progenitor osteoblasts that are lying around to call for more and more osteoclasts, to stimulate more and more osteoclast resorption. And the reason for this is because the skeleton is basically a depot of growth factors and proteins that feed back to the tumor cells and support their growth. So this is called the vicious cycle because it's feed forward when most of our physiological processes are a negative feedback. This is a feedback forward cycle, and it just causes more and more resorption, causes more and more tumor cells to divide and grow, etc. So you can see that this is a problem. So what I'm trying to do with my work is leverage the mechanosensitivity of the skeleton to turn this vicious cycle off. What I'm showing you here is a feedback loop describing the mechanosensitivity of the skeleton. So the skeleton is a load-bearing structure. My training is mechanical engineering, so when I approached this area back in undergrad, starting in the skeleton, I mean, you can view it as any kind of load-bearing structure. The only difference is that the skeleton is alive. So if there's a need for greater strength or greater fracture resistance, this is the process by which that can be taken care of. So let me walk you through this. Bone remodeling is happening in our skeleton all the time. So in fact, we basically get a new skeleton every 10 years. So what that means for us is that the local strain environment, which is the stimulus for this feedback loop, is non-zero. So there needs to be some amount of stresses and strain in your skeleton so that remodeling can proceed as it should. So let's say you increase the mechanical stimulus, and here you can think of maybe exercising, playing a sport, something like that. So your strains go up, which says to this, this feedback then will kick in to increase the bone mass and alter architecture to improve strength, thereby bringing the strains back down to the set point. Conversely, if you decrease mechanical stimulus, and here you can think of being really sedentary, you can think of bed rest or microgravity, well then this remodeling process leads to a net bone loss and reduced strength because, metabolically speaking, your skeleton is expensive. So this is the process by which the skeleton can optimize its structural needs with metabolic costs. So this is the principle that we're trying to use to turn off bone resorption, loss of bone mass that happens in breast cancer metastasis. There are some things we know about what this mechanical stimuli should look like or what the characteristics should be. The number one thing is that this applied loading, the mechanical stimulus, must be dynamic. So if you lift a really heavy weight and just stand there, that's not doing anything for you. You need to move the weight up and down or something like that. You can enhance the osteogenic response, that means the bone-forming response, by increasing the strain magnitude, increasing the strain rate, and increasing the frequency. So these are all ways that we can improve bone formation via the bone remodeling process. And this, I mean, the clinical application for this work has originated in the osteoporosis patient population. Osteoporosis is too much bone loss, basically. So people have looked at exercise or physical therapy quite widely to address osteoporotic patients. So, for example, children and adult women, they respond pretty readily to increases in physical activity by increasing their bone mass. Postmenopausal women, which is probably what you think of when you hear osteoporosis, postmenopausal women, they have a reduced response to exercise, but they can respond. So what this is telling us is that physical therapy or exercise therapies are viable clinical therapy for pathological bone loss, like osteoporosis, and this led us to think, well, what about pathological bone loss due to metastasis? And this is, if you'll remember the cartoon I've been using, this is basically showing you my hypothesis of how mechanical load is going to interfere with that vicious cycle. We know mechanical load stimulates bone formation in the osteoblasts, but we also think that it's going to effectively cut off the food source for the tumor cells. And there's some evidence, some epidemiological evidence to back this up. It's not a lot, but there's some promising results showing that physical therapy has improved the quality of life during chemotherapy, so there's reduced fatigue, reduced weight gain, and it's even had some marginal gains in the bone area, so it's either prevented or even reduced or attenuated bone loss. After therapy, and if you'll excuse me, this should be post-chemotherapy, so after breast cancer patients have undergone chemotherapy, exercise has prevented bone loss and has increased muscle mass. So these are some modest gains, but they're telling us that, again, altering the mechanical environment of the skeleton may be a way to help prevent bone loss or recover lost bone. And we'll table that for a second. So again, this is my hypothesis that increased mechanical loading will inhibit tumor-driven osteolysis and tumor formation in bone metastatic breast cancer. So now I'm going to move into my experimental approach. In my field, we need to have both an in vivo and an in vitro component. So the in vivo component is so that you can use your hypothesis in a complete organism or a complex organism like a mouse. So what I mean by that is, a mouse has a cardiac system, it has a circulatory system, it's got an immune system, etc. But in the mouse models, you can't dissect what's happening at the cellular level. And that's why we have an in vitro system. So that's a cell-based system. So we culture up tumor cells or bone cells or whatever, and we apply mechanical load in a very controlled environment so that we can see what's happening at the cellular level. I'm going to first tell you about my in vivo work. The in vivo loading model that we use is tibial compression. So what you're seeing in the top left is a schematic of our tibial loading device with a mouse tibia in it. The tibia is your shin bone. So we lock the knee down in place, and we deliver cyclic loads through a magnetic actuator through the foot. We're interested in the tibia because, relatively speaking, there's a large volume of cancellous bone at the proximal end, which is the end near the knee. And remember, cancellous bone is 1, the place where fractures usually happen, and 2, where metastatic cancer cells usually colonize. And this picture in the bottom is of a mouse in our loading device. And you can see our waveform in the top right, so you see that it's dynamic. And I'll give you some more details on this waveform in just a second. So this is where I'm going to tell you a little bit about my PhD work. Again, my PhD work was focused on treating osteoporosis. But this model that I'm telling you about, in vivo tibial compression, looking at cancellous bone, is pretty new. So I had to do a lot of characterization first. So one of the studies I conducted was looking at growing mice, because we definitely know growing organisms respond, and we looked at sex differences. So what you're seeing down here are representative micro-CT images of the loaded tibia and the controlled tibia, and you can see right away, you can appreciate, we increased bone mass with the loading. And when we quantified these images, here bone volume fraction is a measure of cancellous bone mass. You can see comparing the solid loaded bars to the open controlled non-loaded bars that we increased bone mass, and when we compared female and male juvenile mice, there wasn't any difference between the sexes. So then I moved into an aging female mouse model, and that's supposed to model aging-based osteoporosis. That's type 2. So type 2 osteoporosis, men and women get this kind of osteoporosis. So we applied tibial loading, and what we saw over here is that, firstly, we could increase mass using our tibial loading device in an aging mouse model. So this is telling us that we can modulate mechanical forces to increase cancellous bone mass. And the second thing we saw is that the response in these these aging mice was reduced. We're looking at the delta between these two bars is reduced relative to what we saw in the young. You just saw this data and this red data in the slide before. But the really interesting result we got was that if you applied mechanical loading in adult mice, we could bring the mass levels up to what we saw in the young. So this is the dashed line here. So this was a positive result, saying that we can really stave off this bone loss with mechanical loading. In touching on the reduced sensitivity, again, we saw reduced sensitivity despite the fact that we were inducing similar tissue strains in the cancellous bone. So we did some finite element modeling, and we saw that there were similar strains among the cancellous bone, but a reduced response. And finally, I moved into looking at tibial compression in hormone-deficient mice. So this is the type 1 osteoporosis that you guys probably think of, the estrogen-deficient kind. And it's a similar story. We increased bone mass with tibial loading, but the cancellous architecture changed relative to whether there was estrogen withdrawal or whether there was loading. I'm happy to talk to you about this in more detail after the talk, but in the interest of time, I'm going to move forward from these results. But again, what this is telling us is that tibial compression is a good model for us to study how mechanical loading in cancellous bone can affect metastasis-mediated bone loss. So for my experiments, I used a SCID mouse model. This is an immunocompromised mouse so that we can inject human cells without there being a rejection of the cells. I inject the cells directly into the tibia, into the marrow space. So this is a model of metastasis that has already occurred. And I have a PBS control group. I'm injecting a human metastatic breast cancer cell line. It's called the MDA MB231 cell line. I don't get bogged down with all what the cell line is called, but I'm going to be using this cell line throughout all of my studies, so I want to point it out so for those of you who are interested, you can know which cells I'm using. So we applied tibial compression daily, Monday through Friday. We applied 1,200 cycles at 4 hertz. We had a loaded group and a non-loaded sham group, and we applied loading for 2 and 6 weeks. So before I could proceed with this experiment, I needed to figure out, well, what's the force that I'm going to apply to the tibia? And the way we do that, the way we select our force, is using a commonly used technique with strain gauges. So we apply a strain gauge to the tibia. This is an ex vivo picture, but we do this while the mouse is alive but asleep. So we apply a strain gauge to the tibia, and if you up here at the knee, we have an inline load cell. So when we apply forces through our device, we can get simultaneous strain measurements as well as load, and we increase the load not over time, but we do this several times with increasing loads so that we can get a microstrain load relationship. And this is how we choose our peak force, by using this relationship. We shot for 600 microstrain. This represents a mouse jumping off of a little tower in its cage. So it's a physiological activity, but it's above that steady state strain set point that I talked to you about before. So again, it's physiological. We're not inducing damage, but it's above what's customary for these mice. And that corresponded to 4 newtons. So we're applying 4 newtons during our cyclic loading. That's our peak force. I looked at bone morphology via microcomputed tomography. I focused on the cancellous bone again in the proximal end, so the end near your knee. And I looked at bone volume fraction. That's a measure of bone mass. I also looked at trabecular thickness and separation, and these are measures that look at literally how thick those little trabecular struts are and how far apart they are from each other. And this gives us an idea of what's the architecture looking like. And then I also looked at histological sections for tumor formation. So what you're looking at here is basically the summary of my results using this mouse model, where if you draw your eye at the bottom, you can see right away that when we applied loading to tumor-bearing bones, this is pretty normal. There's no bone loss, but over here you see. This is serious osteolysis happening, which we prevented totally applying mechanical loading. So this was my hypothesis, but this was a pretty striking result. And when we quantified this, we saw that indeed with mechanical loading, with tibial compression, we increased bone mass. So again, this is comparing the solid bars to the white bars. And this bone mass increased even though there were tumor cells around. So the tumor cells weren't interfering with the response to mechanical loading. And the bone mass increased primarily by thickening of individual trabeculae. And this is a pretty typical loading response. So again, this is telling us that the skeleton remains mechanosensitive even though there are tumor cells around, so this might be a way to address metastatic bone loss.
Okay, if I interrupt you on the previous slide.
Yeah.
Go back in the control.
Yep.
It appears that the non-loaded case has more bone mass than the loaded case. Can you clarify this?
Yes, I think this was just a function of what images I chose. And so it's not easy to get these pictures. But if you look at the quantification, that's not the case, where we did the loaded bones were indeed higher in mass. Yep. But that tells me I should get a better picture. So then we moved on to look at the histological sections where even if there weren't osteolysis, what's going on with the tumor cells. And it was a similar story where this is an H&E stain, which is a standard pathological stain. We had a board-certified pathologist score these blindly. And in a nutshell, there were just no tumors in the loaded bones, in the loaded sections, none that we could see histologically, relative to, again, the non-loaded where there's quite a lot of osteolysis happening. So the histology was corroborating what we saw via micro-CT. And when we looked at those H&E stains at high resolution, we saw that in the loaded bones that there were a lot of activated osteoblasts around, versus in the non-loaded bone, we saw these large osteoclasts over this little packet of bone that they had resorbed next to the tumor that had formed in the marrow space. So, so things are happening like we would expect them to, and we verified the presence of osteoclasts via a trap stain. So for you non-bio folks here, a trap stain is a specific stain for osteoclasts. So we just wanted to make sure what we saw in H&E was really what we thought it was. So the conclusions that we can draw from these in vivo, these mouse studies, is that tibial compression increased osteoblast activation even though there were tumor cells around. And that without tibial loading, there's a high prevalence of osteoclasts on degraded bone tissue adjacent to the tumor. And this is what we would expect in a regular situation where there are cancer cells that we let just do their thing, if you will. We saw that tibial compression inhibited metastatic bone degradation for up to 6 weeks, and I remind you of how striking these results were. And when we went back to literature to try to prove to ourselves that this is really something that this is real, if you will. And there just are hardly any studies around to corroborate these results. Some folks looked at 2D cyclic stretching of a monolayer of lung cancer cells. There is one group who's looking at an in vivo model. This group is looking at a vibrating plate. How many of you may have heard of the vibrating plate to combat bone loss? It's been in it's been in popular press a little bit. They looked at ovarian cancer, it's apples and oranges, but they got similar but not as striking results. So what we need to know now is, well, what's happening to the tumor cells? We didn't see any in the histological sections. Are they dying? Are they going somewhere else? Are they becoming dormant? We don't know. So this is where the in vitro system is really critical for us understanding what's happening mechanistically at the cellular level. So for this section, for my cell work, I have two sections. One is the direct effects of loading, so that's when we directly load tumor cells in our model, which I'll describe to you in a moment. And then we have an indirect effects of loading angle where we load bone cells with tumor-secreted factors around. So is loading affecting the tumor cells or is it affecting the bone cells? We're trying to figure that out. I will tell you right now before we proceed that a lot of this work is still ongoing. In fact, I have students probably running experiments right now. So let me describe to you what our in vitro cell culture model is. Our lab has developed a PLG-based scaffold incorporating hydroxyapatite particles. Hydroxyapatite is the crystal phase of our skeleton. And very briefly, I'll tell you how we fabricate these scaffolds. Basically, we measure out our HA particles and PLG particles and salt. We mix them up and we press them into what really looks like an aspirin pill. And then we put these scaffolds under high pressure, high CO2 pressure, and when we release it very quickly, the HA and the PLG foam together such that the HA is exposed to the surface, so the cells see the crystals like they would in vivo, and then we leach out the salt with water, so leaving behind pores. So the skeleton is a porous tissue. We're trying to recreate that using this scaffold. In previous work in our lab, not done by me, when we seed cancer cells, when we put cancer cells in the scaffold, we see increased survival, proliferation, and then subsequent osteolytic capability due to the presence of this HA. So this is telling us that this is a pretty good model for the skeleton, for metastasis in the skeleton. But I have more work to prove this. So, again, I'm telling you about mechanical forces. So when you zoom in to what's happening to the cells, the cells are the ones doing all the work of bone remodeling or resorption in the case of cancer. When you zoom in to what's happening to the cells, they're experiencing quite a lot of different types of forces. So cells that are attached to the bone surface, as many of them are, when you have strain, so we walk around and we put strains on our skeletons, the cells will deform with the matrix deforming. But then because it's porous, fluid-filled, when you deform a porous scaffold or a porous substrate, you'll get changes in pressure that causes fluid flow, which causes shear stress. And then this also creates ionic gradients because then you have the movement of ions and proteins and things like that. So there's a lot happening at the cellular level. But with our porous scaffold, when we apply compressive forces to the scaffold so that we can recreate all these things that are happening in the skeleton. So the way we do that, we culture our scaffolds with cells in them in a 24-well plate. And we apply force to the specialized platen. So if you imagine I have my scaffold here, I'm basically applying a cylinder directly to the scaffold to create bulk strain. And we do that using a bioreactor. And we do this in collaboration with the Benasser lab, which is next door to my lab, or my PI's lab at Cornell. So to make sure that we're not plastically deforming these scaffolds, I did a little bit of materials characterization, a stress strain a stress strain curve like many of you are familiar with. We chose 10% compressive strain, which is well within the linear elastic range. This was great for us because when you survey literature, 10% strain is pretty commonly used in vitro studies, so this will match up well with what other people are doing. There we go. So we seed tumor cells. Again, it's the same human metastatic breast cancer cells that I used before, the MDA MB231 cells. As I said before, we applied peak strain at 1 hertz. We used, again, a cyclic waveform. We applied this for 1 hour per day for 3 days, and we have a non-loaded group as control. So I looked at a panel of genes related to osteolysis and bone remodeling, but for the purposes of this talk, I'm going to focus on one gene in particular for RUNX2. The reason that this was something that we were interested in, RUNX2, is because RUNX2 plays a significant role in breast cancer bone metastasis. So RUNX2 is a transcription factor that's in many cells, not just cancer cells. A transcription factor is important for connecting external stimuli to the cell. So you get external stimuli to the cell, this transcription factor gets activated somehow, and then it shuttles a message, which is its activation. It shuttles a message to the nucleus so that the DNA can synthesize a new protein. So that's, as I said before, the transcription factor is basically a coupling between external stimuli and protein expression from the nucleus. So RUNX2 in particular induces secretion of proteins that both induce resorption as well as inhibit bone formation. And in fact, when RUNX2 activity was abolished in breast cancer cells, osteolysis was prevented both in vivo and in vitro. So this is telling us that RUNX2 is an important factor in breast cancer bone metastasis. So when we applied 3 days of mechanical loading, we saw, well, the tumor cells didn't die. So what you're looking at the top are representative live dead stains from our scaffolds after they had been loaded. Green represents live cells and red represents dead cells. And you can see there are basically no red cells around. All the cells are alive. Loading is not killing the cells. We also looked at the growth, the DNA content of these cells to get an idea of are they growing. And we saw that their growth profile looked the same. But as you may have expected, we saw changes in RUNX2 expression in the tumor cells. So in fact, we saw that loading reduced RUNX2 expression by about 20-30%. So now we're trying to expand our understanding of what is the mechanosensitivity of this expression factor, of this expression, in response to loading. So what I mean by that is we're trying to basically get some dose response curves or some things like that. So here's one of the first studies we ran where we modulated how long we applied mechanical loading. So how quickly does this gene expression turn off and how quickly does it get back to normal? So what we saw here is that after 30 minutes, we didn't see much going on. At 1 hour, we saw a reduction, but then it was back to control levels at 2 hours. And we're trying to also do things like change the strain magnitude, maybe the length of the number of days rather than the number of hours that we apply load. We're trying to understand what's the sweet spot, if you will, for when this RUNX2 expression goes down so that we can do some subsequent studies on bone cells. So that's ongoing. So I'm going to switch gears now to look at our indirect studies. And for this part, I'm going to talk to you about, I'm going to focus in on osteoblasts. Remember, those are the cells that build bone. Osteoblasts are important because, not just because they're the ones that build bone, so they're the effectors of mechanical loading. These cells, when they differentiate even further, become what's called an osteocyte. They are the mechanosensors. So put that osteocyte in the back of your mind, because I'm going to talk about that at the end of the talk. So again, but for this, we were working with osteoblast cells. So one of the reasons why we focused on the osteoblasts, not just because they're the cells that respond to mechanical loading, but because we don't really understand fully how osteoblasts are operating in breast cancer bone metastasis. We know that progenitor osteoblasts are stimulated to call more osteoclasts to resorb bone, but other than that, things are pretty murky. So for example, progenitors secrete both pro and anti-tumorigenic cytokines. So are they helping or hurting? We're not sure. We've seen that tumor cells have stimulated and suppressed osteoblast differentiation. We've seen that osteoblast activity is reduced and suppressed, and that tumor cells cause apoptosis in osteoblasts. That means cell death. So it's a murky picture. One of the problems is that many of these studies are conducted in 2D. So, we don't live in 2D. Our bodies are not 2D. So when you culture up cells in 2D, it's probably not going to reflect what happens in your body. And in fact, there's a lot of data supporting this. So the power of what we're trying to do is that we have this 3D scaffold system. So we use this scaffold system, but the first thing I needed to do for this project is verify that osteoblasts grow and differentiate and do what they should be doing in our scaffold system. So to prove that, we compared HA versus PLG control scaffolds. And for my osteoblast model, we're using bone marrow-derived human mesenchymal stem cells. Excuse me for not writing that out. Mesenchymal stem cells are the stem cells that give rise to osteoblasts. And in these scaffolds, we have an induced condition where we stimulate chemically the MSCs to become osteoblasts. And then we have a control condition where we do not provide any of these differentiation factors. And we did this, we cultured these cells for 21 days. That's how long it takes for a stem cell to get to be an osteoblast. And then we also include a 60-day time point to see, to make sure that the tissue that is formed is indeed bone tissue. So again, this is, I'm just going to show you a subset of this data. What you're looking at here are mature osteoblast markers. So alkaline phosphatase, it's a marker of a mature osteoblast. So you can just keep that in mind. And when we compared induced, which is the solid bar, so these are the cells that we're trying to get to become osteoblasts, we compare the solid bars with the control, and we see that we can induce stem cells to become osteoblasts just fine in the PLG and HA system. But when we look at long term, the HA system is the alkaline phosphatase levels are persisting. So this is telling us, well, our HA system is better than the control. And when we look at total protein, the reason we're looking at total protein is because, as I said to you before, osteoblasts, their job is to lay down new bone. And basically your bone is mineralized protein. The osteoblasts lay down a bunch of different types of protein, which is then mineralized. So the job of the osteoblast is to spit out proteins. So we wanted to get an idea of what's happening to the protein secretion, and we saw at the earlier time points, HA is secreting more. When we get out to long-term time points, the amount of protein is the same between PLG and HA, but we know that it's mostly an osteoblast-type protein. So this is telling us, okay, in your scaffold system, stem cells become osteoblasts. So when we looked at these things histologically, this is now a Mason's trichrome stain. What we're looking for is collagen. As I told you, osteoblasts, their job is to spit out protein. Well, that protein is mostly collagen. In fact, that's the most common protein in your body. So collagen, type 1 collagen. And when we compare induced to control, we see that there's much more blue in the induced. If you're having difficulty seeing, you can draw your eyes to these arrows. They're pointing out blue spots for you. And we saw more in HA. We're in the process of quantifying this. And when we looked at it 60 days, you can see there's quite a lot of blue in the induced situation, but there's even more in the HA scaffold. In fact, you can see a nice fat layer of collagen on the surface of the scaffold with cells, I'll say embedded, but cells in and among the matrix that's been laid down. So again, this is telling us, okay, this scaffold that we want to use, it's good for osteoblast culture. So we can use it to interrogate, okay, what's happening when you add mechanical loading and when you add cancer. Oh, excuse me, we also did micro-CT of those scaffolds looking for subsequent mineralization. So what you're seeing here is that that matrix that I just pointed out to you, it also became mineralized relative to the control condition, and we quantified that. Excuse me. So now we're ready to add cancer. So we do this in steps. So the next step, we just add cancer before we add mechanical loading. The way we do that is by using what's called tumor conditioned media. This is a way to this is the first step that you use before going to a co-culture system. Putting two kinds of cells in one scaffold is what you want to do, but then it becomes difficult to suss out what cell is doing what. So we start with this conditioned media study. The way we do that is we culture up those breast cancer cells and we let them secrete their factors into the media. We put that media onto the mesenchymal stem cells to see how is this affecting their differentiation. So we have, for our conditions, we have a tumor factors condition. We have a control minus TCM is tumor conditioned media. We have a control condition for that. And again, we're looking at differentiation. So we have induced and control. And we cultured these for 21 days. So what we're looking at here is that, what we're seeing here is that tumor factors appear to enhance osteoblast differentiation. So if you remember, alkaline phosphatase, that's a marker of mature osteoblasts, is increasing with tumor factors in the media. So this is telling us, okay, osteoblasts are differentiating more strongly with tumor factors around. And when we look at total protein, with tumor factors around, there seems to be some sort of modulation in how much total protein is being secreted when there are tumor factors around, but we're still trying to understand what this means. As I said, this work is ongoing. So now we're going to look into loading. So this is the final part of my project. This is the part that's still going on now. So if you'll forgive the lack of answers I can provide you regarding this. So we have a loaded condition, same loading that I talked to you about before. We have a non-loaded control group, and here we're again adding tumor factors, for the tumor conditioned media that we got from the breast cancer cells, and a control group. And all of these cells are receiving osteogenic media, which means we are feeding all of the cells factors to try to stimulate their differentiation. And what we see here is that loading is modulating the effects of how tumor factors affect osteoblast differentiation. So here, again, don't get bogged down with what osteocalcin is. It's a marker of a mature osteoblast. So here we're looking at gene expression. What we see when we look at osteocalcin, which is a late-stage osteoblast marker, is that with loading in the control condition, we get an increase in this expression. That's what we would expect, right? Because osteoblasts become more bone forming under conditions of load. So this makes sense. When we apply tumor factors, this goes away. So it's unclear if we miss the window, if you will, or if the response is delayed. We're trying to figure this out. And when we look at this factor, this is RUNX2. It's the same factor I told you about before. But remember, I told you this is present in all of our cells. In osteoblasts, it's an earlier stage marker of differentiation. So when we asked ourselves, did we miss the window or are we too early? We looked for this to try to get a handle on what the timing of things are. And basically what we're seeing is that with tumor factors around, this expression is going up. So this is telling us that with loading, with tumor factors around, loading is increasing osteoblast differentiation. So the conclusions that we can draw from in vitro studies is that mechanical loading did not directly affect tumor cell viability or growth, but instead reduced expression of RUNX2, which has a downstream effect on remodeling. Effects of altered RUNX2 expression on this downstream osteolytic protein secretion and changes in bone remodeling still needs to be verified. So we have not proven that there's a link between these reductions in RUNX2 and bone remodeling, but that's what we're working on now. And we saw that what I think is happening is that both loading and metastatic cancer stimulate initial osteoblast differentiation, but what loading is doing is that it's ensuring the survival of mature matrix-producing osteoblasts even though there are tumor cells around. But as I've tried to tell you, osteoblasts have they have a long differentiation process, and their function is different at each stage of this differentiation, so it's a complicated problem that the field doesn't quite have a handle on. So overall, we see that mechanical loading inhibited the growth and osteolytic capability of secondary breast tumors in bone, and I again remind you this result that proved we should be paying attention to the mechanical environment in cancer. And these effects were not due to loading-induced tumor cell death directly, but were instead mediated via decreased expression of genes that interfere with bone homeostasis. So as I think about, so what does this mean? What am I going to do in the future? I've been focusing on the osteoblasts, and if you remember, I told you to keep your brain tuned into this type of cell, osteocyte. So to recap, these are the results that we're working with. This is proving to us, okay, mechanical loading matters. The mechanical environment matters. And I'm interested in the osteoblasts. If you remember the osteoblasts, they're the effector cell in that feedback loop. But the osteocytes are the mechanosensors. So I'm showing you a cartoon of an osteoblast laying down bone. Well, when these cells are done laying down bone, they become osteocytes, which this cartoon is trying to convey to you. And they form, they look like neurons, don't they? They're dendritic. They have projections reaching out to the cell next to it. These osteocytes they make up 95% of all the cells in the skeleton. They have this incredible connected network. They connect to each other. They connect to cells in the marrow space. They connect to the vasculature. They connect to everything in the bone, and they are basically running the show. But we have no idea what role they play in bone metastasis. But we do know that these cells are also mechanosensitive. They are the mechanosensors. So we want to utilize the mechanosensitivity of the skeleton and specifically this cell to understand how can we help improve patient outcomes for breast cancer metastasis patients. So this is the same cartoon. I just described this cartoon, so I'm going to blow right through this in the interest of time. But I would like to take a moment to acknowledge my postdoctoral advisor, Claudia Fischbach, as well as many people in the Fischbach lab who helped an engineer get up to speed with cell biology. It was a steep learning curve, but with lots of help, I did it. And buried in there are also some of the undergrads who helped me complete a lot of this work. I'd also like to thank my PhD advisor, as well as our collaborator, Larry Benasser, who helps with the bioreactor studies, as well as some other folks who helped with analysis along the way, and of course my funding sources. So I'm happy to take any questions.
I realize this research, at least the impression I get, is that it's fairly exploratory in terms of using mechanical loading to control or influence metastasized cells and so on, growth. But you mentioned something about sweet spot, which to me is code for optimization. And I think the in vivo studies seem to be more conclusive in what the loading was doing. And you were loading, I forget what the load was, but it was at 4 hertz. And so there are some numbers there that refer to the mechanical aspects of what you're doing. The question is, how does one get at, say, an optimization of load? Frequency is kind of 4 hertz is kind of a fast oscillation for loading for what we normally experience.
Sure. So yes, I hear what you're saying. Let me tell you very quickly where the 4 hertz comes from. The 4 hertz is the stride frequency of a running mouse. So that's why we chose 4 hertz. When we moved into the in vitro system, we chose 1 hertz because that's the stride frequency of a person, and that's very commonly used in cell culture work. There's oftentimes a disconnect between the parameters we use in mice versus the parameters that we use in cells, because having a system that recapitulates in vivo is we're still working on that. That's a whole field of research. So there's often a disconnect. Okay, so you're talking about optimization. I mean, the real answer is that when the term I'm going to use is mechanotransduction. How do mechanical signals turn into cellular signals? And we don't know. So we have some ideas of what happens to the cell, but we don't really know what's happening. So when we think about optimizing, honestly, we're trying to reach a threshold where we can induce bone formation and go from there.
Nice work, nicely presented. In the process of your exploring these responses, did you bump into the issue of piezoelectric behavior in bone and collagen as a possible means of transduction?
So I'm familiar with. I know that occurs in bone, but we did not look at that at all. But I know it's an effect. So I showed you my cartoon of what happens to the cells. I zoomed in on a cell. The short answer is there's not a whole lot of information on how the piezoelectric effect is contributing to bone remodeling.
Because there was a period of time in clinical orthopedics that electrical induction of bone healing
Right.
was rampant, and it seems to have just pretty much disappeared in the clinical arena.
Right. But if I remember my homework, there are some folks here that are looking at that, right?
Yeah.
So is there any new news that I should know about?
No, it just seemed to disappear.
Yeah, you're not the only one who's commented on that to me. There are still some believers who think that an electrical signal is the way to go. But you're right, there's just not a lot of that work being done right now.
If I could pursue a follow-on, as a clinician, we look at x-rays with osteolytic lesions in them and cringe, especially when we think about mechanical
Yeah.
stresses applied. How do we see this research work transitioning into the clinic to get
Right.
our patients vibrating or whatever we're going to do?
Right. So, I'll hit on that first. The vibrating plate, it may be a way to deliver low strain magnitude signals to the skeleton. So, I mean, it's a problem in the osteoporotic community too, where you get an elderly population whose skeleton is already fragile. You can't tell them to go lift a bunch of weights, or not usually. So the way I think about this translating to the clinic, I mentioned the vibrating plate, but there's a lot of other things that are coming online where I think about the timing of when you apply physical therapy may be the way to go. So before huge amounts of osteolysis have occurred, you should be using physical therapy. So for example, circulating tumor cells is a hot area as a biomarker of what's happening. After you have a resection, there are still cells around, but we don't really know what's happening, why things recur, where they go. So circulating tumor cells may be a way to combine, to figure out when should we introduce physical therapy, something like that. I think probably physical therapy is good no matter what after you have cancer, because for example, I mean, I showed you all that work with the quality of life stuff, but also subsequent inflammation is a real big issue. It's correlated with recurrence, with cancer recurrence. So if you could help keep your stress levels down via exercise, you might have some gains there. But really, this is an open area. I'll go with you. Okay.
I saw you show a figure on comparison of half an hour, 1 hour, 2 hours,
Mm-hmm.
and the same is most effective at 1 hour. So I was wondering, if you try this another day, just continue the experiment. Is it always the case, or is there a physical reason for that?
Both. So we have not run that experiment yet, but my answer based on what I know is yes, we would still get that the next day. The reason for that is that these cells, like most cells, if not all, they desensitize to mechanical loading. They desensitize to stimuli. So there is, the cells will respond up to a point, and then you need to give them a break. So some of that, we don't totally understand why, but here's an example. So mechanical loading, we don't totally understand mechanotransduction, but we know that, for example, things change to receptors. So maybe you get a downregulation of receptors with mechanical loading, so they're not gonna respond anymore because they're all downregulated. But if you give them some time, then the receptors will become responsive again, and you can apply load. And there's lots of in vivo data to support what I just told you, where if you give them a rest, then they respond.
Maybe I can give you more work to do. Once you do that experiment the next day, but you do it again the next day and the next day. In training for a marathon, your body adapts and you get more fit, and you can run for longer and longer. I'm wondering if that half hour, after you've been doing this regimen for a couple months, maybe that half hour now stretched to an hour or 2 hours or so. That might not be a fixed point. Like it might have to do with how in shape the mouse is.
Sure, but remember, if you remember the feedback loop I showed you, let me go back to it. There's a target strain. We think it's a strain, but there's a target strain that the skeleton is shooting for. So if the osteoblast gets stimulated to lay down new bone, it's to reduce the strain profile in that local area. So once the osteoblasts do what they need to do to bring the strain environment back down, then presumably they won't respond anymore unless you keep increasing the strains. So, if you're training for a marathon, there's also this idea that your body gets used to just running. So when you go swim, you're really terrible at it. So, there's an accommodation that's going on even at the cellular level.
You're injecting human breast cancer cells into immunodeficient mice to make your experiments. How reasonable is it to think that this is representative of something that would go on in a human patient with cancer, for example?
It's a big limitation of the field. So people want to have a humanized or human model. So we use immunocompromised mice, but you're right, the immune system, a fully functional immune system, plays a role in cancer. It's a limitation that we all struggle with. So that's also why these 3D in vitro models are so desirable. There's some. So yes, your point is taken that the translation to what would happen in humans, we have to be careful when we think about that.
When you set the strain for the in vivo experiment, it looked like you had maybe a 0.1% strain, and then on the in vitro you were up at might have been 10%.
10%.
Remembering the numbers, and the duration I think for the in vivo was maybe 5 minutes.
Yep.
You went up to much longer durations. Would you comment on
Yes.
why those are so different?
Yes. This may also require some more detailed talking after, if you really want to know. So you're right. There's a disconnect between what happens, what we have to apply in vivo to get bone formation versus what we have to apply in vitro. As you mentioned, there's an order of magnitude higher strain. So the reason for that basically is that in our skeleton we have a natural amplification system where we can take the very small strains that we put in our skeleton that get amplified to something that cells respond to, which would be on the order of 10% strain. I'm happy to tell you how that works. I think I may need a piece of paper to show you.
Sounds like an interesting mechanical analysis.
Yeah, and it's at the level of the osteocyte. So the osteocyte, if you're looking at these cells, they look dendritic. Well, they're sitting in these little tiny tunnels that are filled with fluid. So you deform the skeleton, and there's fluid moving around, and these osteocytes are attached to the matrix in ways at a scale that I'm not showing you here. And all of that stuff, that architecture contributes to the amplification process.
there are no questions, I invite you to speak to meet with the speaker, and I'll bring this to an end. Thank you.
Thank you.